இது போதுமா இல்ல உங்களுக்கு தெரியுமா? ஓகே ஓகே. இது ஓகே. இது வந்து போர்டு வந்து
Good evening, ma'am. Hello, ma'am. Good evening.
மீட்டிங் விட்டு லெஃப்ட் ரைட் வரக்கூடாது. இல்ல மீட்டிங் விட்டு லெஃப்ட் ரைட் வந்தா கரெக்டா
Lady.
Namaste, sir.
Hello, ma'am. Good evening, ma'am.
Am I audible, sir?
Yes, ma'am.
Yes, sir. You can start.
Just a minute, ma'am. Let me present the questions that the students try to answer in cell cycle and cell division.
Okay, sir.
Yes. Yes, sir. Mail வரல. Fantastic. Hello, students. So, good evening, all of you. So, hope just check. Can you all able to see my screen? Okay. The screen is visible for you. My screen. Yes. Okay. Now, I'm just presenting the question paper one after other, and I request all of you to work out for the questions and note down your answers. And after the session, maybe I will be sending you the answer key. You all can verify your answers. Okay? Any doubt? Hello, students. Can you hear me? Hello, ma'am. Are you there? Hello, madam. Are you there? Students, if you can hear me, just raise your hand. Yeah, okay. Yeah, then ready for the test analysis? Yeah. Okay, first question. See, all of you take a paper or notebook and answer the questions one after other. Okay, please settle fast.
Sir, screen is not visible, sir. The shared
Screen
screen
is-
is not.
Screen is not visible, ma'am.
Yes, sir.
Oh, just a minute.
It was, now it is not visible.
Just a minute, ma'am.
Take it, ma'am.
Hello, ma'am. Is it visible now? Hello? Okay. Madam, now is it visible?
Yes, sir.
Yeah. Yes, students, please. Just 25 questions are there. Okay. I think we can work out those 25 questions. Very simple thing only. I request all of you to just write the title, cell cycle and cell division, and then put the serial number from 1 to 25. Like one after another, you can answer the questions and note down your options. And later, the answer key will be sent. You can verify it. Okay. And if you're done with the first question, we can go to the next question. Yes.
Yes.
So tell me, or shall we read? Which of the following is present in maximum number in adult cell? What type of cell number is high in adult cell? Since it is, yes,
Cell, sir.
what?
Somatic cell, sir.
Okay, okay, good. And all of you can also just work it out. Yes, next question. The sequence of cell cycle is
Option B. Option B, sir.
option
B.
B.
G1, yes, G2 and M, sir.
Okay, option B. Fine. Next, synthesis of RNA and protein takes place in which phase of cell cycle? Hello, students. Hello. Yes, tell me for question number 3. Hello. Synthesis of RNA and protein takes place in which phase of the cell cycle?
Yes, sir. Sir,
Yes.
M phase, sir. Option B.
Did anyone give the answer? Someone is telling? Yes, someone is coming to answer. Hello, students. I-
Option. Sir, can
Yes.
you hear me, sir? Option C, sir.
Oh, okay, good, very good. The next question. Fourth question. During which phase can nucleolus be observed clearly? You can consider mitosis. In mitosis, during which phase nucleus be observed clearly? Nucleolus, not nucleus, it is nucleolus. Hello, students.
Option B, sir.
Option
B, sir. Early prophase.
oh, early prophase. Okay, is that so? Any other option? One second. Any other option? Okay, fine. Please note down this answer. We can check after your after the session. Just a minute. Okay, next question. See, question number 5. Which structure can be observed at the surface of centromere during metaphase?
Option A, sir.
Option A.
Yes,
Which
sir.
structure? See, listen. Which structure can be observed at the surface of centromere? I think option A you are telling, right?
Yes, sir. Yes, sir.
Okay, very good. And question number 6.
Option D, sir. Both nuclear and cytoplasmic.
Mitosis actually means
Option D, sir. Both nuclear and cytoplasmic.
okay. What is the technical term for nuclear division? And what is the technical term for cytoplasmic division? Yes, karyokinesis and very good. Karyokinesis and cytokinesis. Okay, very good. And then question number 7. Sir, battery loan வருது. Yes, sir. Okay, question number 7. The role of mitosis is not merely to divide a cell into 2 daughter cells, but to ensure genetic continuity from one cell generation to another. And the mechanism ensuring this genetic continuity is what?
Option A, sir, formation of two cells with identical DNA.
See, you read the question carefully.
Yes, sir.
What is the role of mitosis? Are you telling? First one now, is that your answer? Okay, very good. I think someone said option A.
The loss of
Okay, fine. Next, question number 8. The stage of mitosis during which?
Sir, excuse me,
Yes,
sir, kindly discuss the answers for each one.
okay,
Proper reasoning, sir.
okay, madam. With this, for from the beginning?
Yes, sir.
Okay, madam, okay.
Actually, I just started with the interface.
Okay.
Still prophase only, I have thought, but in between this 1 week was a gap for them. I couldn't catch them. As it was a unit test assessment mark entry was there. So let us start and we need to explain each step, please, sir. If the
Okay,
question was,
madam.
we need to
All
analyze
the
and
questions
answer.
with explanation.
Yes, sir.
I don't have to ask the answer for them, right?
Yes, sir.
Okay, madam, fine. Thank you.
I
I'll
doesn't
stop.
know the topic. Only mitosis prophase, still that only I have thought to come.
Oh, mitosis
I
prophase
couldn't
only.
complete.
Okay.
Yes. As
Okay,
for
okay.
last 1 week, I couldn't catch them. I couldn't complete the topics.
Okay, that's fine, ma'am. I'll take it. I thought it would have over. Fine, ma'am. I'll take it. Okay. Yes. Okay, students. Fine. See, we are. The first question is about what? We are multicellular organism. Actually, this topic is about cell division. Okay. And of course, we have in our body, we have two type of cell, right? One is somatic cell and another one is what? Reproductive cell. Okay, somatic cell and reproductive cell. So, based on that only in adult body, okay, like adult person, we have that just see all these things like B, C, D, like gametes, like reproductive cell, same thing only. If fertilized, it becomes zygote. So, as you said, this is the correct answer. So, correct answer, I'm just marking it in orange. Okay, somatic cell. Okay, next. The sequence of cell cycle, actually, like, the cell cycle and like finally, here only the mitosis comes and it starts with what? As you know, G1 apart from Z0 and then S and then G2 and then M. Okay, so that is the perfect order. Okay, G1, S, G2, M. I think you have given that answer correctly and fine. Next, the third question is synthesis of RNA and protein takes place in which phase of the cell cycle? Okay, so listen, in a cell cycle, okay, in a cell cycle, for example, look at here. For example, this is S phase. Okay, this is S phase. In S phase, what occurs? The replication of, listen carefully, in S phase, what occurs? The DNA replication occurs, synthesis of DNA. Okay, so for the synthesis of DNA, like what are all the enzymes and protein molecules required? Okay, the enzymes and protein molecules required are synthesized here in G1 phase, and then only like the DNA replication can take place. Okay, so that's the correct answer. And of course, you people give me the correct answer for that question. Okay, like what is the synthesis of RNA in which place of the cell cycle? It is actually G1 phase. Okay, next, during which phase can nucleolus be observed? Okay, during which phase the nucleolus can be observed? Here, and I asked you to consider mitosis. Okay, see mitosis. Mitosis and meiosis are there actually. So, if you want, take mitosis as an example, early prophase, you can see in prophase, actually the disappearance, only you can see disappearance of nuclear membrane and the other organelles you can see. Okay, in metaphase, actually the chromosomes are aligned in the center. Okay, and only here you can say, okay, appearance, reappearance of nuclear membrane, reappearance of all other organelles. Fine. Okay. And then see the questions which we pass through, we just go fast. And if you cannot able to answer, means understand anything, if I miss something, you please let me know. Okay. And question number 5. So, which structure can be observed at the surface of the centromere during metaphase? Which structure can be observed at the surface of the centromere during metaphase? See, when you look into the structure of chromosome, see, look at that. When you look into the structure of chromosome, see, this is the basic structure of a chromosome. And then here, this is actually called what? Constriction. They call it as what? Primary constriction. They call it as what? Primary constriction. It is called what? Primary constriction. And centromere. We call it as what? Centromere. Okay, centromere. And here in the centromere, there is a disc-like protein is present. Okay, in the centromere, there is a disc-like protein is present. That is called what? Kinetochore. Okay, that is called what? Kinetochore. Okay, kinetochores are the one which is present in the centromere. Okay, and in metaphase, see what is the basic structure of a metaphase chromosome? Basic structure of a metaphase chromosome, it actually looks like this. Can you understand? So, it actually looks like this. And both of the chromosome, listen carefully. So, both of the chromosome actually are connected with what? There are two chromosomes. See, this is one chromosome and another one is called what? The duplicated copy of chromosome. It's called what? Sister chromatid. Okay, the duplicated chromosome. So, both of the chromosomes have a share a same structure, which is called what? Kinetochore. Can you all able to understand? Hello, students?
Sir.
Yes, let me repeat. This first one is actually the the orange, dark orange one is the structure of a chromosome. And only during metaphase, you can see the duplicated copy of the chromosomes. That's why I have given here in a shaded form. Okay, duplicated copy of the chromosome. And both the chromatids basically are connected with what? The kinetochore protein which is present in between. Okay, and the special aspect of this kinetochore protein is there only. See, listen, what is the importance of this kinetochore proteins? There only the spindle fiber can come and attach. Okay, you know, the spindle fibers can come and attach to that kinetochores only from all the ends, the spindle fibers, and then they pull apart the chromosome. Okay, for the segregation of chromosomes, this can occur. Okay, so please be sure about the structure of chromosome, centromere, kinetochores, etc. Fine. Now, what is this question? Mitosis means many options are there. Why you ruled out this? Because no division of means reduction division occur. This is the answer thing for my meiosis. Only in meiosis, reduction of number of chromosomes. For example, 46 to 23, 23. Only in meiosis. But in mitosis, you can see 2n become 4n and then 2n and 2n. Okay. So, mitosis, only parent cell produces daughter cells. So, for this to occur, in between two important events are, as you said, karyokinesis and cytokinesis. Okay, nuclear division is called what? Karyokinesis. And then cytoplasmic division is called what? Cytokinesis. Okay, fine. That's the sixth question. And then seventh one, the role of mitosis is not merely to divide a cell into two daughter cells, but to ensure genetic continuity from one generation to next generation. Means you can directly come to the thing in the sense mitosis ensures genetic continuity in what? Formation of, that is the main thing actually. Parent cell produces daughter cells and identical to each other. That is mitosis. Parent cell produces daughter cells. One parent cell produces two daughter cells which are identical to each other. Okay, and which ensures genomic level identity. That's why option A is correct. Okay, fine. Okay, so question number 8, I think from here we should start first. Now you please answer and then we will go for the discussion. The stage of mitosis during which the nucleolus disintegrates and chromosome appear is known as what? Now you just answer, then let me give the explanation. Hello students. For this question, please answer. Question number 8. Someone come and answer, then we go for the explanation. The stage of mitosis during which nucleolus disintegrates and chromosome appear is known as what? Hello students, can someone answer?
Yeah, prophase, sir.
Very good. Okay, prophase. In prophase, okay. Okay, like the disappearance of nuclear membrane as well as before, like actually the condensation of chromosome is appears. Okay, before that, how the chromosome is present? The chromosome is present in a bead on the string model. Okay, beads on the string model. Okay, like this only before cell division. Okay, before cell division, the chromosome appears like that. Okay, only during prophase. Okay, only during prophase, the chromosome condenses actually. Okay, condenses to form what? Chromosome. Okay, this is called what? This structure. This is chromatin. What is the structure? They call it as what? Chromatin fiber. Okay, chromatin fiber condensed to form what? Chromosome. Okay, condensed to form what? Chromosome. Okay. And one simple question let me ask. Why it is called chromosome? Hello, students.
Yes, sir.
One simple question. Why it is called chromosome? See, we call it as chromosome. How many chromosomes we have? What are the types of chromosome in human? Hello, students. Okay, listen. Normally in a cell, okay, when a cell is in normal state, inside this is actually the nucleus. You just consider this as a nucleus. In this form only, the DNA is present in chromatin fiber form, which is not in condensed form. Okay, only so you cannot see that. Okay, it is a kind of diluted one. So you cannot see that. Only during cell division and the chromosome, actually the chromatin fiber condensed to form what? Chromosome. Okay, and if you add any stain, okay, if you add any stain, you can see chromosome. Okay, you add some stain or dye which gives color to the chromosome. So that's why it is called chromosome. And see, one more question let me ask. What stain they add to visualize chromosome? Do you know that? Chromosome can be visualized by adding what stain, what dye is used? Dye or stain. What stain is used? Note down, sometimes they may ask, they will be adding acetocarmine. Okay, note down the dye, which is called what? Acetocarmine, which gives a kind of red color to the chromosome. Of course, kind of red. Okay, orangish red color to the chromosome. Okay, or red color to the chromosome. Is it clear? Fine. So keep that in mind. And next. Fine. Ninth question. Can someone come and answer? Chromosomes can be counted during what phase? Please, someone come and answer. Chromosomes can be counted during what phase? Prophase, metaphase, anaphase, or telophase?
Beta phase.
Telophase.
Beta phase.
Ah, okay, fine. Metaphase. Okay, very good. What is this technique called? Do you know about the technique? See, counting of chromosome can be done by a technique called what? Note down. Karyotyping. Very good. Karyotyping. Okay, the number of chromosome can be studied. And what is the number of chromosome we have? How many number of chromosome? In humans, n equal to what? Listen, n equal to what?
What is it, sir?
No, see, 2n equal to 46. Up n equal to what? 1n. 1n equal to what?
20. Sir.
Yes,
23.
1n equal to 23. Okay, fine. So that can be done. And do you know any disorders like this? For example, sometimes maybe in your genetics you study, in some people, maybe they have 47 chromosome. For example, X, X, Y, a kind of sex chromosomal abnormality. In some people, they may have 45. So it is called XO syndrome. Okay. So if the number of chromosome changes, that results in genetic disorder, chromosomal abnormality. Okay, keep that in mind. If the number of chromosome in an organism changes, that results in chromosomal aberration. So, counting of chromosome can be done by this technique, by which we can able to identify abnormal number of chromosomes. Okay, fine. Now, question number 10. The nuclear membrane disintegrates and spindle appears at what? Hmm?
Option D, sir.
Option B,
Late prophase.
see, late,
Prophase, sir.
okay. You know the events of prophase, metaphase, anaphase, telophase?
Yes, sir.
Okay, you're telling late D then, what did you tell?
Sir, option A, sir.
Oh, you said option A. Okay, fine. See, listen, what is the function of spindle fiber? What is the function of spindle fiber? See, what is the function of, I think I told you little early, if this is the chromosome and centromere, okay, the spindle fiber, let me give, what color? Another different color. See, the spindle fiber attaches here to the chromosome and they basically pull apart the kinetochores. Okay, the centromere, the kinetochores, they come and attach and then they pull apart the chromosomes. Okay, fine. So, metaphase only, in the state of metaphase only, the chromosomes are aligned in the center. Okay, aligned in the center of the cell. Okay, so this is nuclear membrane disintegration, prophase. Okay, and then spindle fiber should start appearing. Okay, you can say because all these phases are basically seriously it occurs one after other. Okay, it starts with later prophase and of course metaphase. Okay, fine. So pro metaphase, okay, fine, can be considered. Next question. The separation of two chromatids of each chromosome during early anaphase is initiated by. And before answering this, what are chromatids? What are chromatids? Hello, students, what are called chromatids? Listen, this is the chromosome and the duplicated copy of that. Okay, the duplicated copy of chromosome. Okay, the duplicated copy of chromosome is called what? Chromatids. Okay, so two chromatids. Now, the separation of two chromatids of each chromosome during early anaphase is initiated by. Yes, what are the options?
Option B, sir.
Yes, please check all of the options.
Yes, sir. Option D, sir.
Option D. Okay, means all of the processes are connected with each other. So it's a series of process which supports the pulling apart, the karyokinesis. Okay, fine. Next, the telophase stage of mitosis is
Option D, sir.
option
D, all of the above.
Okay. The lost state of karyokinesis, telophase. More or less opposite to prophase, absorbed in cytoplasm. Okay, fine. So, all of the above. And please keep this in mind. PMAT. PMAT means what? Prophase, metaphase, anaphase, and telophase. So, just keep that in mind. And then karyokinesis is used for. The term karyokinesis is used for.
D. Option D, sir.
Are you telling B or D?
D. Sir, overall changes occurring in nucleus during the cell division.
Okay. See, karyo represents what? Carry on means what? Nucleus. Kinesis means what? Division. Okay. Yes. Fine. Okay. Overall changes occur in nucleus during cell division. Basically, nucleus divides. For karyokinesis to occur, before that, what should occur? DNA synthesis should have completed. Okay, DNA synthesis means replication must be completed. Then only karyokinesis can occur. Okay, fine. Next question. How many mitotic division must occur in a cell to form 10 to the power 24 cells? See, always apply 2 into n. Just calculate and tell me.
Sir, 64, sir.
Oh, if 64, all of you got the same thing. Okay. See, cycle 1, 2. So 2 to the power n, they basically say. Okay, fine. So all of you, please, just a minute. Okay, fine. Good. Next question. The difference in the division of plant cell and animal cell is? Hello, students?
Yes,
The
sir.
difference in division of plant cell and animal cell is? See, in plant cell, see, listen, when cytokinesis occurs, for example, in animal cell, from outside to inside, cytokinesis is initiated. Okay, in animal cell, okay, the outside to inside cell division is initiated from the membrane to the inner region in animal cell. But in plant cell, actually, if this is the cell, from here, it should start and calcium means the cell differentiation is initiated. Okay, like this. Okay, so here in the equational plane, basically it occurs. Fine. Then
Sir, option A, sir.
for which one you're telling?
15th, sir.
Okay, during which stage of meiosis crossing over occurs? During which stage of meiosis?
Yes, sir. We didn't learn, sir. We don't know meiosis, sir.
Oh, you have, see, listen, in meiosis, prophase 1 is very, very important thing. Okay. Just prophase 1. That's a very important thing you should know about meiosis. That prophase 1 consists of what? See, keep this in mind. L, Z, P, D, D. In short form, it is called what? L, Z, P, D, D. The order of processes which occurs in prophase 1. Okay. First, L is leptotin. L is what? Leptotin. And then Z is called what? Zygotin. And then P, pacatin. Pacatin. And then diplotin. And then diakinesis. Okay, so these are all order is very, very important. This order is very, very important in meiosis. Okay, in leptotin, the chromosomes started to pair with the homologous chromosome. Okay, the homologous chromosome. In the first stage, the homologous chromosome started to pair with each other. That is the first thing in leptotin. In zygotin, okay, leptotin, in zygotin, what forms? Like they come closer to each other, like this, they come closer to each other basically. Okay, and along with what? The tetrad formation, they call it as what? The tetrad formation. It occurs. Okay, the homologous chromosomes with its duplicate sister chromatids. Okay, and then the crossing over is very, very important. Let me make another important thing. See, for example, this is chromosome, and this is sister chromatid. They call this one as what? Sister chromatid. And actually, the crossing over occurs between the sister chromatid. So, this chromosomes, this is sister chromatid, this is sister chromatid. I just give a new color for that. And in pacatin, these sister chromatids, okay, only exchanges the chromosomes, basically genes. Okay. Okay, and here only this chromosomes exchange the genetic material. So genetic, what forms? They call it as what? Crossing over. Crossing over occurs between, okay, this particular thing. Okay, crossing over. So pacatin is the very, very important thing. So chromosomes come closer to each other here, and then homologous chromosome pairing come close to each other, pairing occurs here, crossing over occurs here, longest phase actually. And then they call it as what? Synaptonymal complex. Synaptonymal complex, and crossing over occurs, which results in what? Recombination. Yes. Okay, just keep this in mind. And then diplotin and diakinesis, they just separate each other with the shuffled, genetically recombined chromosomes. And keep one thing in mind, this chromosome and this chromosomes are non-recombinants. Only the sister, non-sister chromatids are recombinant chromosomes. Okay, fine. So the next thing, at which stage of the following, means in which, just a minute. Okay, proteins, microtubules required for mitosis. In which stage? Different kind of proteins and microtubules required for mitosis. This is connected with mitosis. Where it is synthesized. Already we saw RNA and protein synthesis, but here For mitosis, what is in which phase this molecule should be synthesized? It should be synthesized in G2 phase. Okay. And then if the initial amount of DNA is denoted as 2C, then it increases to what? In mitosis. So, for example, 2C, then it should become what? 4C, and then 2C, 2Z. Okay. So, not this one, 4Z. Initial amount, it is increases into 4Z, and then here DNA should replicate first in cell division, and then only the parent cell produces two identical daughter cells. So you can give this as an example answer. And now 19th question. During interphase, chromosomes are recognized as chromatin network. Okay, this is the statement. Chromosomes are highly dispersed during this phase. Is this correct or not? Both, assertion is during interphase, chromosomes are recognized as chromatin network. And reasoning is chromosomes are highly dispersed during this phase. What will be your answer? Can someone tell me? Hello, students. Assertion and reasoning. Hello, students.
Yes, sir.
Yes.
Sir, option B, sir.
Option
B, sir, B.
B. Okay, correct. Means during interphase, actually chromosomes are recognized as chromatin network. Fine. See, here chromosomes are recognized as chromatin network. Chromosomes are highly dispersed during this phase. You cannot say that actually, because if it is condensed, then only it is called chromosome. Okay. So they should not use this term chromosome or highly dispersed. No, it is actually only chromatin fiber. If it condensed, then only the chromosome forms. Okay, just one more question before you leave at least. So, just a minute. Okay. In A stage, centromere duplicated. And then statement Q. In the prophase, centrioles separate from each other and move towards the opposite pole. There are two statements.
Okay.
Just hello? Want to tell something? Hello?
Yes, sir.
Yes? Yes? Can someone tell me the answer? And let me ask you one question. In which stage centrosome is duplicated? That is the direct question. Okay.
Option.
Hello? See, when centrosome is duplicated.
Option A, sir. Option A, sir.
Option A,
And Q both are
statement
correct.
P and Q are correct. Do you think so? Okay. See, actually, before the DNA replicates, okay, before the synthesis phase, In prophase itself, please keep in mind, in prophase itself, sorry, in yeah, actually in prophase only the centrosome actually duplicates. Okay, so in centrosome is duplicated. In the prophase, centrioles separate from each other and move towards opposite direction. Okay, means
Sir,
Yes.
excuse me, sir. Sir, this is a time for dispersal, sir.
okay, ma'am. Okay, students. Thank you.
Thank you, sir.
Yeah, thank you.
हाँ